The one thing that all rare disease communities have in common is their indomitable spirit. This year's Unsummit is living proof of that. There's no denying how much we miss seeing everyone in person, but at the same time, we're determined to make this year's virtual event an event we will always remember, full of new connections, insights, and ideas that are going to take us to new heights of achievement. Rather than limiting us, this year's virtual Unsummit has expanded our reach, allowing us to connect to more global audience members than ever before. Right now, we are streaming in from 29 different countries around the globe. We're working together to make this a better world for all of us. COVID-19 has tested our resilience, but it hasn't stopped us. To help foundations in a lean fundraising year, we have launched our Continuity of Care Fund and Continuity of Research programming. These initiatives have helped organizations continue to support patients and families as they confront this added challenge to health and well-being. In the midst of this public health emergency, we have worked to maintain public awareness of the fact that 400 million people are affected by rare diseases. We must stay united around our commitment to the #1 public health issue in America and the world by supporting education and vaccination around the COVID-19 virus, by protecting our priority status for continuity of clinical trials, for calling for greater access to gene-based diagnostics at all ages, beginning with newborn and pediatric onset diseases, and by continuing our gains in telemedicine when it's a preferred pathway for supportive care. We're also committed to being champions of the greater inclusion and bridge builders for people of color who have suffered from systemic racism and institutional bias. We believe that healthcare is a fundamental human right, independent of culture, religion, race, nationality, and politics. We began this year's Unsummit with a week full of one-of-a-kind events. First, we heard from our future leaders, young adults talking about self-advocacy and mental health, followed by a 3-night watch party with the Disorder Channel and Sunday night's Rare Impact Grant Concert fundraiser to support families through the COVID-19 crisis. Next, we will be bringing you a packed agenda of presentations and panels available via our event platform. Today, Partners in Advocacy and Innovation Stephen Kingsmore from Rady and PJ Brooks from NCATS will be interviewing Sonia Vallabh, patient and researcher at the Broad. They are kicking off a day of exploring how we as advocates can be leaders in science and ensuring inclusive research and being the bridges to underserved communities. I want to give you all a special reminder that each night we will have community programming around wellness, self-care and family support, and the human side of being a researcher. I'm especially excited as a rare mom myself to have Hollywood star Sam Humphries join us for an evening reading of You're Full of Genes. That's geared to the whole family, and it's at 7 p.m. Eastern Time and 4 p.m. Pacific Time. Tomorrow, we go deep into the science, exploring the opportunities in artificial intelligence, machine learning, and how the processing of this data explosion we're in can be harnessed to improve access to diagnosis and treatment for everyone. Wednesday will feature keynotes and panels on gene therapies and other potential therapeutic modalities and the innovations in clinical trials that rare advocacy leaders are helping to drive. On Thursday, we'll be talking about access to critical therapies and the role patient advocates play in improving access. From improving access to diagnostics to being heard in value-based pricing to becoming a proactive financial self-advocate will make the inaccessible language of hospitals, insurers, and policymakers real. Actionable impact packaged for our communities to lead. And to close out the week, we will make sure we all circle up around the word that has built this grassroots community, this family, hope. We'll feature some great sessions on staying connected through these times, expanding our family, the diversity of our voices, and then close out with a special keynote to remind us all that this is also very doable.
Hello, everybody. My name is Stephen Kingsmore, and I'm the moderator for this next session, together with my co-panelists, Sonia and PJ. Welcome, everybody, this Monday morning or Monday afternoon, depending on where you're at. We're very excited to be talking to you today and to sharing some insights in terms of what we are doing to make your lives a heck of a lot better. So what we're going to do is, first of all, we're going to hear from Sonia and then myself and then PJ, and then we are going to ask you for questions. So if you would start to enter any questions that you have as we talk, we will then pick those up in our chat box and respond to them for the remainder of the talk. I hope you got that. So let's get started. It's my pleasure to introduce to you Sonia Vallabh, who's going to tell you her story. Sonia.
Great. Thank you so much. Thank you, everybody, for having me today. And I will be talking about some of the science of trying to treat or prevent genetic prion disease, but also some of the personal side of how I got into this work, because it's been an unusual road. So our story begins in 2010. That's the year I went through the heartbreaking experience of watching my mom die totally unexpectedly. So she began that year as a healthy, beautiful 51-year-old. And by February and March, she was having what seemed like mild and miscellaneous symptoms, a little bit of trouble with her eyesight. She lost some weight unexpectedly, not for any reason we could discern. But this snowballed in a way that was just terrifying. It was like watching a time-lapse video. By March, April, May, she was losing her ability to communicate, recognize us, do sort of her normal daily activities. She was losing so much muscle that she couldn't move around the house safely. And by June, she was on life support. So I think what we saw was, a rapidly progressive dementia. But at that time, I didn't have the words for what I was seeing. All I knew is that she was dying. And it was at the end of that year that she passed away without a diagnosis, even though we had sort of rushed her around from hospital to hospital in search of one. It was only from her autopsy that we learned that she had died of genetic prion disease. And in that word genetic was sort of the whole next chapter of this adventure, which I had never suspected would have a next chapter. I realized I was at 50-50 risk of having inherited the mutation that had caused this horrible disease, genetic prion disease. I, together with my husband, Eric Minical, decided to get predictive genetic testing, got the report, and you can see it right here. It was not the result we hoped for. I was positive for her mutation. At this time, we really didn't have scientific background to sort of process this news. I was trained as a lawyer. Eric was trained as a transportation engineer. But the day we got the test report actually ended up being this turning point in a way we never could have anticipated. We found ourselves super motivated to learn more about prion disease and what was being done. At first, the goal was just to be savvy patients because we felt this disease was too rare for us to be able to rely on doctors to tell us if something was developed along the way. But that goal evolved, and we found ourselves drawn into the process of learning more, leaving our old careers, and retraining as scientists from the ground up. So today, we lead our own prion lab at the Broad Institute, and we're working on therapeutics, but also all of the sort of supporting pieces that will make, I hope, therapeutics meaningful once we have them. And I'll say a little bit more about the ways in which this journey has been surprising, not just the journey into science, but even the journey since we got here. So this, at a very high level, is the blueprint of prion disease. Forgive me for showing some science cartoons, but I think what this says to me is that at its heart, despite all the mysteries, prion disease is actually really simple. And I think this is where we have a real advantage as a disease caused by a single known gene. The prion protein gene gives rise to one RNA, which gives rise to this one protein, shown in blue, that we all have. Normally not a problem, but it's able to misfold in a way that is extremely troublesome. And that's where you get disease. So this was sort of the blueprint that we came to when we got into science. We sort of said, okay, here are the different points where we can think about intervening. And originally, our focus was kind of on the normal prion protein. because there was good evidence that if we could reduce the amount of that protein, that should be protective. So this is a gain of function disease. It affects the brain. So we thought, okay, can we lower the amount of that protein in the brain? I think maybe the biggest thing that we have sort of, the biggest way our thinking has evolved, the biggest thing we didn't appreciate in the beginning is that going after that protein was going to be a kind of from scratch effort. And I think this is an interesting thing about proteins, right? They are often the sort of effectors of disease, these molecular machines, but each really has its own shape corresponding to its function, and each is sort of its own problem. So we did launch a whole program to try to find molecules that would bind the normal prion protein and maybe inactivate it, and maybe we could use them to degrade it, or some other useful goal. But I'll sort of give you the punchline. That program 6 years later gave us no useful chemical matter that bound to the prion protein. It's just a really, really hard problem. And I think, maybe our protein is particularly hard in some ways, but I think we're not alone in that. The place where our quest really took an unexpected turn that has been just super, super useful and super productive was us sort of getting our heads around the idea that maybe the protein was not the right place to go after this disease. And that if we could think about moving our focus upstream, each protein may be different from the next. But if we look at the DNA or the RNA, we see more commonalities. We see more shared structure, more shared chemistry from gene to gene. And it was a tremendous watershed for us, realizing people are creating technologies that target the DNA, that target the RNA. And if we can identify tools like that have worked for a different gene, maybe we can repurpose them in a way that will be useful for our gene. So this is just a schematic of some such tools, and I won't go into detail on all of them. I know we'll be hearing from Dr. P.J. Brooks in a little bit, and he may shed much more light on the sort of amazing landscape of platform technologies and how it's evolving. For us, it ended up being antisense oligonucleotides that seemed like the tool that was most applicable and most realistic. And it's now been 6 years since we first launched our collaboration with Ionis Pharmaceuticals to look at ASOs that target the prion protein RNA and maybe reduce the amount of RNA in the brain so as to reduce the amount of the protein. And here we've seen progress at a kind of a pace that I never could have imagined when we first got into this. By using a tool that already had proofs of concept in the human body and in the human brain, that was already being used to reduce the levels of other sort of disease-associated RNAs, we've really been able to sort of piggyback off of existing knowledge to drive this drug development program forward. And we're now at a point where we hope to be in the clinic in the next few years. So from sort of being handed a diagnosis 10 years ago, getting into science 8 years ago, I don't think I could have foreseen that I would be saying that to you today in 2020. One thing I want to touch on is how our role has kind of evolved as we have sort of embraced these platform technologies as maybe our most feasible path forward. We got into this saying, we need the molecule, we need the molecule, we need to discover the molecule. And reorienting our quest around these genetically targeted platform technologies has really meant for us realizing that the molecule may actually come out of partnerships with the right people who are working on the right technologies. And in fact, our role might be better described as making this rare disease partnerable, making it ready for partnership. So what has that meant for us? It's meant assembling all of the tools kind of on a platter that are needed to make companies and other kinds of partners come along and say, even though there isn't a large patient base in prion disease, we think that there's good evidence that we can make a difference here. And we see that everything that we're going to need to launch a drug development program is there. maybe some of these are things that a company would do for a common disease, but we really came to realize the scope of our job was much, much broader than we had thought on day one. So to give just a couple examples, we realized it was up to us to work on a biomarker that could read out drug activity in a living patient. That was something that people were going to want to see. We realized that it was up to us to work on quantifying risk as best we can. Some of these things, you know, we looked at the field naively in the beginning and said, oh, we have that, right? We have mouse models. But the devil was really in the details for all of these. And for mouse models, it turned out how many of those models have the whole gene? And if you're talking about an ASO that might target any part of the RNA, you really need the mouse to have the whole gene. So we ended up developing new mice. And then in terms of patient engagement and recruitment, we sort of came along and said, you know, there are lots of referral centers that are expert in prion disease. So probably that's how we'll recruit patients. That piece is probably solved. But in fact, as we've done more and more and more animal studies, we've come to realize that a meaningful piece of the puzzle is going to be trying to prevent disease. This is a disease that moves so fast that may be the most good we can do, is to get to people at genetic risk before symptoms even start. Now you're talking about a whole different form of recruitment. You're talking about trying to gather people who are perfectly healthy today, but have a genetic mutation that puts them at risk. And this is something that hasn't been done systematically in our disease. So it's something that we're now trying to do. So those are just a few examples of how our job has really expanded from what we thought it was going to be. But I think even though it has required us to sort of learn a whole bunch of new skills to try to build up this arsenal, it's been worth it. It continues to be an uphill battle every day. I think we still have a lot of work ahead of us, but I feel hopeful about where we are. I feel kind of amazed that we have, you know, a plausible therapeutic pathway ahead of us, and we'll do whatever we have to do to walk it and try to make a difference. With that, thank you so much and wish us luck.
Yep.
Thank you so much, Sonia. I hope, audience, that you're like PJ and I, you're just amazed at the resilience and ingenuity of this lady. What an amazing story to go from being a patient to being an inventor and somebody who's aspiring to solve their own disease. I hope that resonates with you, and I hope for some of you it excites you about what you can do, what you can truly achieve, even though you may seem right now like you're in a low place. I hope that Sonia inspires you. Now, we're going to come back to Sonia in a little bit. Keep typing in your questions. Thank you that you've started. But first of all, a couple of minutes from me and a couple of minutes from PJ, because we're going to lend some additional perspectives. So as I said at the start, I had a research institute. It's called Rady Children's Institute for Genomic Medicine. And so Sonia's focused on prion disease, and our focus is on about 10,000 genetic diseases. The simple genetic diseases, they used to be called Mendelian diseases. We're partnered with the third largest children's hospital in the US, Rady Children's Hospital in San Diego, which is where I'm talking to you from. And our focus has been on ending the diagnostic odyssey. You're very familiar with this, that long wait between a child or yourself having symptoms and the physicians knowing exactly what the triggering cause of those symptoms are. And for many diseases, that's the gateway to specific treatment, what we call genomic or precision medicine. In Sonia's case, that's not true yet, and she's having to go that extra step to actually come up with innovative therapies that will impact the course of disease. But for us, we're focused on this initial problem, which is how do we connect a patient with an answer? And so we've come up with a method of doing that. It's called whole genome. And this seems pretty simple. We decode the entire human genome code in every single patient, and hopefully also in mum and dad. Now, the genome is a big space. The genome is the instructions for being a human being. It's the blueprint of, and it's 6.5 billion letters long. And it's a mistake in one of those letters or one of those sections of the code which causes a genetic disease. We've been able to reduce the time to decode the genome and assign a diagnosis to 36 hours. Can you imagine that? That it's now possible today to telescope the diagnostic odyssey to 1.5 days. So we started this about the time that Sonia started her journey, so about a decade ago. And today we already provide this to about 50 different hospitals spread all the way across North America, everywhere from Miami to Seattle and from Boston to Hawaii. And so that's our mission, is to end the diagnostic odyssey and to make what we do currently for 50 children's hospitals be the standard of care for every doctor's office and for every hospital when a child or an adult is admitted and might have a genetic disease. That's pretty exciting, isn't it? So if you've got questions about that side of things, also put them in and address them to me. But next I want to introduce you to PJ Brooks. What he's doing is really also amazing. He's in a remarkable part of the National Institutes of Health, and I'll let him talk about the new program that he's leading.
Thank you, Stephen, and it's wonderful to be here with you and Sonia. And I'll be talking a little bit of some thoughts about therapies for monogenic diseases from our perspective at NCATS, and think about what the future may hold for these. So I think we're all familiar with the basic problem of the incredible and increasing number of rare diseases and diseases with known molecular basis. It's going up quite rapidly. The somewhat depressing part is at the present time we have treatments and therapies for only a small fraction of these, about 500 or so. And at this rate, it's going to take far too long to develop therapies for all of these diseases. So you really need a whole different way of looking at the problem. And from our perspective, we actually don't think about thousands of rare diseases. When you really think about it, for single gene disorders, what used to be called Mendelian disorders, there really is a much smaller number of underlying causes, underlying etiologies. You can think of there's two. There's those where you gain a function, like in Sonia's case, or loss of function diseases, recessive diseases. And there's really only a limited number of mutation types. And we call these nonsense mutations or premature stop codons. Missense mutations that cause abnormal protein folding, again, as Sonia mentioned, abnormal RNA splicing and dominant gain of function diseases. And I'm not going to go through the details of this for lack of time, but the important point is that for each one of these, there's a potential therapeutic strategy. There are drugs, for example, you could use for mutations that cause premature stop codons, oligonucleotides for dominant diseases, or siRNAs, for example. And so really, from the perspective of accelerating clinical trials, we like to think of that as that really these causes, these are really the diseases. And what we think of now as individual diseases are different clinical manifestations. And to really get the most people into clinical trials faster, we're ultimately going to need to think about designing these trials differently. But you might be wondering, if you get a genetic diagnosis, where do you get this information about these underlying causes? And in fact, it's probably there. What most people think about when they get a genetic diagnosis is the diagnosis of the disease. But somewhere buried in there, there's going to be information that looks like this, written in a kind of a code. And that's the information about the individual mutations, and also the information about how they can be grouped into these different therapeutic strategies. So one of the things we're most excited about at NCATS now is a couple new funding opportunities we have that are focused on supporting clinical trials of drugs that target shared molecular etiologies in multiple rare diseases. So rather than doing clinical trials one disease at a time, focusing on these etiologies and grouping patients that way. We have one more for academic investigators and one for small business investigators. These are the funding announcements here. If people are interested, happy to talk more about it. And I think this is an important strategy to bring treatments and therapies to many more patients more quickly. It's basically doing what they do now in oncology, which is rather than developing treatments for cancers that affect the breast or the liver, for example, they develop treatments for cancers that affect certain genetic signaling pathways and group patients across different target organs. And that's actually led to drug approvals at the FDA. And I think we can apply that to rare diseases. But thinking even more broadly for monogenic disease, there are some that we still don't have single approaches to, like insertion deletions, but gene therapy and gene editing are broadly applicable to those, to monogenic disorders. And I think one of the things I'm particularly excited about is that looking to the future is the real potential for genome editing for the treatment of genetic disease, and I particularly highlight this work that was published earlier this year from David Liu's group of a genome editor, a molecule that can potentially correct up to 90% of human disease-causing mutations, a single biologic. And if you can imagine this being developed and brought to clinical trials and applied to as many patients as possible, based, again, not on their disease, but on the underlying mutation, I think there's a real opportunity there. And I'll be happy to talk about those and some other therapeutic options and other things we have going on at NIH during the discussion. So I'll stop there now.
Thank you, PJ. So, audience, we got 3 folk who would love to hear your questions and answer them. So I'm going to ask the first question, both for Sonia and for PJ. Let's start off with Sonia. What is the biggest bottleneck right now, Sonia, that's impeding your progress?
Gosh, that's a great question. I think the biggest place where we've had to do some major reorientation of how we think about moving forward to the clinic has been in thinking about the right kinds of clinical trials for prion disease. And maybe, this will resonate with other folks as well, because I think particularly in rare disease, thinking about how to do the right trial is tricky. It's, you're inherently dealing with a low N and the idiosyncrasies of the disease. And in our case, the idiosyncrasy is that symptomatic disease moves so rapidly that it's very difficult to intercept people on that downhill, even with the best, we think, genetically targeted therapeutic. I hope it will be possible someday. But what we saw, for example, with my mom is that she didn't even get a diagnosis in her lifetime. In the meantime, we have predictive genetic testing that can identify this population of people at risk. So in addition to thinking about how to do the best trials we possibly can in symptomatic people, I think we also have to be figuring out how to do trials basically for the first time in these pre-symptomatic people. And I think the bottleneck there has really just been that this is a relatively novel way to think about doing trials. To put a new drug into perfectly healthy seeming people may understandably seem very risky and really isn't business as usual. I think in any disease where you have a sort of early symptomatic phase, there's a temptation to treat then and see how you do. But in prion disease, I would argue we really don't have that. We're catching people sort of as they're dying. So I think we have this mandate to try to be bold and try to prevent if we possibly can. And that has meant sort of thinking about how to recruit these folks, thinking about what we're going to measure in people who, you give the drug and you're not going to see an improvement in symptoms because they don't have symptoms right now. So we're going to have to rely on biomarkers and how can we establish that we really trust them. So I think there's been quite a lot of work to do in that area. But I hope that it's going to be more broadly relevant than just for us, because we are just one example of diseases where you can identify those mutations before anything's going wrong. And of course, the ultimate dream is going to be leverage that genetic knowledge to prevent disease, not just, chase after it.
Thank you, Sonia. PJ, you're at a really exciting point now where you have a strategy and a branch of the government, the NIH, who's endorsing this new strategy. What for you is the biggest bottleneck that you're facing in seeing this strategy advance?
Well, I guess there's a couple bottlenecks to talk about. I think a general one, and that's the goal of this new program, is to get beyond thinking about one disease at a time and thinking about commonalities based on underlying mechanisms. That's a very different way of thinking about disease. The typical situation when a patient is diagnosed is they look at that individual disease and they go to Google and look it up and go on from there, thinking about the underlying cause and the possibility of grouping and being involved with people that have a similar underlying cause. It's a very different way of thinking, and I think it's scientifically it makes sense, but for the community to understand it, we have to start doing these trials and just change our way of thinking, and that's obviously what we're trying to do with this new program. But I also think in terms of genome editing, I mentioned, there we really have some amazing breakthroughs in these new enzymes that can literally edit the DNA and correct the mutation in the cell. And if you can correct that mutation in the cell at the right time of development and treatment in enough cells, you could treat, cure, potentially prevent the disease. The big limitation there is that being able to deliver these genome editors to the right cells. And another program I'm involved with at the NIH that I coordinate as part of the NIH Common Fund is the somatic cell genome editing program. And that one, we've got a total of 20 different projects that we're supporting, all focused on different ways of delivering genome editors to the relevant cell types in the human body. And because we think that's the biggest limitation, is how do you get these to all the cell types you have to correct? And we're also supporting some work like that for small businesses at NCATS as well. Delivery, 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 as they say.
Thank you. And I've got one more question. And this time I'm going to start with you, PJ, so that I don't put Sonia on point again. But same question for both of you. This is a really exciting new time. It's a time of fresh hope for folk who have felt genetic diseases may have been hopeless. So we're excited. But give us some idea of the time frame, if you would. What do you believe will be fundamentally different, PJ, in 5 years and 10 years as a result of this initiative?
Wow, I would like to think that the whole way we're thinking about enrolling patients in clinical trials will be fundamentally changed. Part of the diagnosis will be, here's your genetic diagnosis, but here's the mutation, and based on your mutation, you can enroll in this clinical trial, and those will be available and ongoing. So the time from the diagnosis to clinical trial startup would be a lot less. In terms of genome editing, I would love to think that, there again, some of these platform technologies will be available to many more patients, and it will be as routine as, I don't know that gene therapy is routine now, but we've got a couple approved therapies, and I think that those kind of trials will be quite common, particularly for those tissues where we can deliver the genome editors. And ultimately, I hope we'll be able to deliver them to every cell in the body.
Thank you, PJ. And the same, Sonia. Where do you think you'll be 5 years or 10 years? You can pick. But what will the world look like for for folk who have this curse of potentially prion disease?
I hope it's going to look really different. I very much hope in the next few years we're going to see the first prion protein lowering therapy hit the clinic. I very much hope it's going to hit the clinic in the form of a trial in healthy people at risk, and that we will be testing this drug for the ability to prevent or delay disease, in addition to the ability to treat disease. And from there, the sky's the limit. I think the first drug may not be capable of reducing PRP to zero, but that's my dream. And it's going to be really interesting to keep an eye on technology as it keeps moving. And here, I have to give a shout out to PJ and everything that he's doing on the delivery front. For us, looking at a whole brain disease, the question is always, how can we get the drug of interest to every neuron in the adult human brain, or almost every neuron? And right now, that just takes a huge swath of exciting technologies off the table for our disease. But as we think more and more about delivery to all of these different cell types of interest, I hope that that's a technological hurdle that we're going to clear, and it would open up amazing possibilities for us and for a lot of other disease communities. So I'm excited about where we're headed.
Thank you. And I'm going to answer my own question. On my side, the next 5 years are going to be pivotal. Right now, we have scalable methods to end the diagnostic odyssey for every child who has a disease of unknown etiology. They exist. It's now just a matter of making those available and educating clinicians so that they know, when they don't know the etiology of disease, to order a genomic sequencing test. That's all it's going to take. And I would predict within 5 years that this will be the standard of care. Obviously, the big gap right now is that these technologies are available in tertiary and quaternary referral centers, but not yet in most hospitals and most clinics. And that's what we need to do. We need to get the word out. We need to democratize this so it's available for everybody, every child, every adult who's suffering from a disease for which the doctors don't yet know the etiology. Okay, we're going to move to some of the questions that have been put in over the chat. Some of these are a little tough, and I'm going to have to paraphrase them, so bear with me. So here's the first one. As research moves to platforms, how can we best work together to add a platform approach to approvals that 90% of, and this is talking about drugs, 90% of review is generic and 20% is disorder specific? Is that possible? PJ, do you want to try and answer that one?
Yeah, and I think it is. We work a lot with our colleagues at FDA and the Center for Biologics and also the Center for Drugs. We've talked to them about these different ideas, and they've generally been very supportive, both in terms of the platform approaches for rare diseases, for example. And the fact that that approach has led to FDA approvals in the oncology space kind of illustrates that it's possible. It's just really a matter of adapting that oncology approach for rare diseases and different etiologies, and figuring out how to do that is a main goal of our new funding opportunities. And I think on the biologic side, we work very closely with Peter Marks and are in fact developing some new opportunities along with the Foundation for NIH Accelerated Medicines Partnership for Gene Therapy, that is designed to sort of change the approval process for some of these platform approaches. And I think the FDA is very much open to that. I think it would make their life a lot easier, to be quite honest. They're really being overwhelmed with these things, one disease at a time, and they see the benefit also. So I think there's a lot of opportunity and interest there, and certainly something we will continue to work towards.
I think it was a great question, and certainly the platform approach has revolutionized diagnosis. This idea that one test can test for just about every single genetic disease, same platform, same type of interpretation process. You just plug in the patient's medical record information and you use that to sift through the genome. This has completely changed the approach to diagnosis. So I do think it's a great question. Let's see if we can find another one. Okay, I'm going to just read this one out. It's not so much a question, but it is a question. So this is from one of our audience. I have 4 daughters, 2 with a very rare genetic disease. One's a carrier and one that hasn't yet had genome sequencing. How do I find out if we could possibly be part of one of these collaborative studies or research? We've seen doctors at NIH, CHLA, that's Children's Hospital of LA, and Amsterdam. Let's each answer that. Sonia, for a family out there who have this threat of prion disease, who should they get in touch with?
Sure. So for prion disease, we actually have one centralized center in the United States. It's the National Prion Disease Surveillance Center in Cleveland, Ohio. And they do all the testing for prion disease. It's really an amazing service. So this is testing for someone who's symptomatic, if you're worried that they have prion disease. It's also predictive genetic testing for someone who may be at risk. They do autopsies to confirm diagnosis, and they also sort of serve as a tissue bank for the whole field for research. So they're really they combine an amazing set of different resources. And for families that are, concerned about potential risk or impacted by prion disease, I think it's a good place to start.
Give us that more time, Sonia.
Sure. They're the National Prion Disease Surveillance Center, and they're in Cleveland.
Super. I won't make any Cleveland jokes. I'm being very disciplined. PJ, people want to find out about platform approaches to drug development. How do they get involved? How do they get to be in your loop?
I think right now we're starting to do these clinical trials. I think also, though, as people get information on their the mutations that cause their disease, there are people working on the development of treatments and screening molecules for that, including at NCATS and other places. I think it's hard to give a general answer to that. It sort of depends on the specifics of the mutation. But if whoever wrote that would like to send me an email, I can probably talk to you about that and see what additional help I can provide. I'd be happy to.
Thank you. And in terms of the testing piece, reach out to our organization. It's radygenomics.org, and that's a website. You can find out more about what we do on that website, and there's a way to get in touch with us via email or phone call, and we'd be delighted to see what we can do for you. Okay, I'm going back to the questions again. Give me a second. This is an interesting one. It's from somebody in Gitanjali. I hope I didn't get your name wrong. What's the future of collaborations with countries like India? How can genetic medicines be created for global access? And this isn't just true of India, in fact. We have the same issue in the United States, where we have people who are within driving distance of a tertiary or quaternary referral center, and then we have the entire rest of the country for whom they may as well actually live in India for lack of availability of some of these technologies. So I'm not sure who's going to have a go at answering this. How do we get this available for everybody. PJ.
I guess I'll take a shot at this in to say that I'm part of an organization called URDRC, the International Rare Diseases Research Consortium, and obviously we work with other funders and other rare disease research advocates and researchers from all over the world, including India. And part of what we do is to try to address some of these issues. I can't say whether we've got it all figured out right now. It's obviously a big question, but I can say that it's one that is on the topic of things that we work on at URDRC, and will continue to do so.
Thank you. That is a good answer. That is a marvelous organization. But, you know, this is a difficult question. And unfortunately, in the early going, sometimes these revolutionary new technologies are not very democratic. And sometimes they even increase the distance between the wealthy and the not so wealthy. And this is a tragedy, but we need to recognize it and we need to fix it. I got another question for each of us. Are there public policy hurdles which are hindering broader adoption? And this is true both of whole genome sequencing and also of drug development, either for prion disease or using these bundling approaches that PJ has mentioned. Anybody want to take that one?
I guess I'd a bit toss this one back to you, Stephen. But as I think about some of these therapies and the potential, then I look at newborn screening. It seems to me that given the way that these therapies are being developed now, something needs to change there, because the number of patients that could potentially benefit from some of these therapies is far greater than we're going to be picking up by newborn screening. And for some of these diseases also, because we don't have newborn screening, when you find the patients, they're already symptomatic. And what you'd really want to do, particularly for progressive degenerative diseases, is intervene before the symptoms. So I think some type of change in the way we think about newborn screening would be valuable, but you probably have some thoughts on that too, Stephen.
Yes, I'll have a go at it too.
Okay.
I think that patient advocacy is critical. That's why I put my suit on this morning. I don't normally do that on a Monday morning in this COVID-19 era, because you have such an important role to play. You don't know this probably, but there's actually a bill in Congress. It's not going to get heard this session, but it's called diagnostic. And if our legislators move on that, we can indeed have public policy so that every patient has the right to a genomic sequencing test. What do we need to push that across the line? We need everybody to be calling their representative and encouraging them to support legislation, because right now this is a big bottleneck. And the only way sometimes to get policy bottlenecks removed is for us all to get on the phone and to get writing letters and to let our elected representatives know that this is darned important to us and we want them to fix it. Okay, we've got time, I'm told, for one last question. And so this is coming from Nathan P. And this is a really interesting question. Sorry, and just shouted in, please restate the name of the bill. It's called the Ending the Diagnostic Odyssey Act. That's an easy one to remember. And it's both in the Senate and the House in the federal government. Okay, from Nathan P., and we're each going to take a go at this. How do we know, and this is talking from a patient perspective or an advocacy perspective, how do we know, or what do we look for in an institution that shows they'll be ethical with our data?
I mean, I can jump in. I don't know if I have all the answers. I think it's an interesting moment for that question with a lot of sort of fast-moving pieces, because the amount of data that we can sort of collect and analyze and store has gone up by so much recently, and what we can do with it has gone up by so much. I think that from the research perspective, there certainly are a lot of controls built into the system, and we found that as we've launched our study of healthy genetic prion disease mutation carriers. We went through a sort of tremendously long approvals process to make sure that we were going to treat them properly when they were on site, and treat all of their samples and all of their data properly long beyond when they were on site. So I think my experience on the research side has given me faith that there certainly are controls built in. Now, those controls need to evolve as what's possible evolves. And I think that's a race that everybody is trying to run right now. That is, it's certainly a challenge, but I think there is a lot of good faith in what I've seen of the research world so far.
PJ, do you want to take a look?
I guess I could just say that for research that is funded by the NIH, there are requirements for data security and ethics around the protection of human subjects' data. So that is something to keep in mind.
And I would just add to this a little bit. I think it's a super question. And as you've heard from both of our panelists, there are laws, there are regulations. So that's sort of the basal layer. But I do think, we want to go deeper than that. I got a couple of suggestions for you. Number one is look at that institution and ask, do they have a parent or a patient advisory council? Are they listening to patients and advocates? Do they help them make decisions? Are they helping to oversee policies and procedures? I think that's a really good guide. Do they have an advisory council of lay people? And second of all, how transparent are they? Do you know how they spend their money? Do you know what proportion's going to administrative costs rather than to actual real research to help patients? Send them email. See if you get a response. Those are two really good ways, but I think it's a critically important question that unfortunately sometimes research can be a business. And as patients, we need to ask difficult questions. Are these guys really working in my best interests? Be aware. Anything else to add? Last comments for each of us. We've got just a couple of minutes. What would you like to leave this audience with, Sonia?
Actually, in my last couple minutes, I'll just jump in on the public policy question that we touched on earlier. I was thinking about what are sort of the public policy roadblocks for us. And I think a big one is whether we support people to learn about their risk. When I was, first grappling with the fact that my mom died of a genetic disease, I talked to a lot of people, both formally and informally. And what I heard was, walk away from this if you possibly can. Don't get tested because there's nothing you can do. Even from sort of the formal genetic counseling apparatus, I heard a lot of, well, this is going to make your life difficult. Think about the insurance implications. We don't even know what all the insurance implications could be. And, I sort of knew about myself that I was on one end of the spectrum and I really wanted this information. And even for me, there were just a lot of hoops to jump through to get it. Now I think about people who are sort of maybe more in the middle of the spectrum. They're interested maybe, but they're also scared, which I totally understand. We're nearing a place where having your genetic information in your hand might make all the difference. Having it in your hand before you're sick might make all the difference. And I would love to sort of push the envelope on the culture of predictive genetic testing, the culture of fear around I totally understand people's concerns about insurance, about medical discrimination, about like many of the other implications. But I think we talk a lot about the potential negatives and the downsides. And I want to make sure we give equal airtime to the upsides. Because one of my big fears going forward is, what if we have a preventive therapeutic, but we can't get everyone out of the woodwork because we can't convince people to get tested?
Thank you, Sonia. PJ, a closing comment, please.
Yeah, I think, really focusing on that when you're getting a genetic diagnosis, there's a disease that is written down, and then there's the underlying causes, those mutations, and the realization that, in some cases, with a little girl named Mila that you might have heard about with an antisense oligo, she has Batten disease, and a form of Batten disease for which there's no treatment. But because one of her mutations was a type of abnormal splicing mutation, that underlying mutation was amenable to an antisense oligonucleotide therapy. So thinking about what is a disease, what is an underlying cause, and what is actionable can be really important.
Thank you both very much for being willing to spend time with us this morning. Thank you, Sonia, for sharing your story. It's inspiring. I hope that all of you who are listening realize that this is an unparalleled time. This is a time of excitement when finally we're starting to have at our fingertips technologies that can make a difference for your rare disease. So I hope you leave this morning invigorated, because the world is changing fast and for the better. Thanks so much for joining us.
Thank you.
Thank you.